I'm Na'ama Geva-Zatorsky. I've opened my lab here at the Technion almost 3 years ago at the Rappaport Faculty of Medicine, before which I was at my postdoc at Harvard Medical School in the lab of Professor Dennis Casper, where I was fortunate to make my dream come true, which was to study the gut bacteria, the small organisms, each one cell, that communicate between each other and complement our physiology. And here in lab, we pursue these studies, studying the mechanisms of these interactions, which microbial molecules are important for our health, and how do they communicate with us. So I'll show you my lab. This is the most important room, which is the room where we grow the bacteria. The gut bacteria are anaerobes, so they grow without oxygen. Oxygen actually kills some of them. So this is why we need to grow them in this special chamber, the anaerobic chamber, where we wash out all the oxygen, and we can grow the bacteria in different types of conditions. And here is an example of a tube where you see the media is not transparent, meaning the bacteria grew in it. Otherwise, it would have been transparent. I will also present you to my students, so we can start here first with Haitam and Niru. Haitam is studying the interactions between the gut bacteria and the brain. It's a new field which is called the gut-brain axis, and this we're doing in collaboration with Professor Asya Rolls, also here at the Faculty of Medicine. And Haitam and Niru are studying how can we fluorescently label the gut bacteria to visualize them live, both to study the interactions between them and how they complement the host. You can see bacteria in blue, in green, and in orange. And these are 3 different bacteria labeled with 3 different colors. You can see them simultaneously while they're living, and it can be also visualized inside live organisms. This is Maxime, who has recently joined the lab. And this is Rawi, who is studying several projects. Rawi is studying how the gut bacteria can be used as therapeutics for cancer, both to cancer prevention and for cancer eradication. And together with Nadav and many other in the lab, they led recently a project on corona, actually, where we developed a protocol to detect corona in people. It's actually now developed as a home kit. So pink is negative and yellow is positive. And the idea is to spit and visualize in several minutes whether you are positive or negative for the virus. Nadav and Rawi also study antibiotic-resistant bacteria. You can see here a plate with bacteria that are antibiotic-resistant. So it's a special plate where these bacteria grow black. So you can count them, and after the method that they developed, you can see that the microbes, these antibiotic-resistant microbes are eradicated. And the last bay here is the bay of Lilach, Noah, and Dana. Lilach and Noah are studying how the gut bacteria communicate with the immune system, and the actual interactions, the actual molecules, and the crosstalk between them. And Dana is studying the viruses in the gut, the bacterial viruses, so viruses that infect bacteria. How are they important to bacterial functions, and how does this affect the host? And the last room is this room, which is called the tissue culture room, where we grow bacteria together with host cells, mainly the cells from our immune system, sometimes cells from our blood, sometimes other cells, and that's how we can study the actual interactions between the bacteria and the host. So all in all, we study how these gut microbes, which are called the forgotten organ of our body, can complement our physiology. In which disease states are they important, and how can we harness them as therapeutics? I would really like to say it's very exciting to talk with you. Any of this work would have been possible without your help. It's very crucial to us. And this Technion environment is a really amazing place to work at. A lot of collaborations, a lot of expertise, engineering, biology meet together and really work together on projects. I really feel we're at the cutting edge of science worldwide. Thank you very much.